Dr. Hansen here. I hope that working through that glycolysis gave you some insight into how to think about enzyme nomenclature and regulation. I don't expect you to be able to write down the entire sequence of reactions, but if you're given an enzyme name, I hope you could interpret it some to understand the substrates and products, what type of reaction occurred, especially for the enzyme names which are used over and over and over, such as kinase, phosphatase, dehydrogenase, isomerase. Such names you absolutely want to interpret, and then you simply look at the name before the enzyme, pyruvate kinase. That's something you should absolutely know. Hopefully you do have some feel for how glycolysis works, because glycolysis is the oxidation of glucose down to pyruvate to make ATP, and gluconeogenesis is the buildup from pyruvate to glucose in order to regenerate glucose, and it costs a bunch of ATP. Accordingly, many of the enzymes are shared, and the language which describes both processes can be understood similarly. Accordingly, I have the same review learning objectives for the gluconeogenesis talk as for glycolysis, and even these new learning objectives on gluconeogenesis parallel the learning objectives for glycolysis, in that they are focused on enzyme nomenclature, regulation, the DAM model, and on energetics. Does it go up or down in energy? Also in parallel to the glycolysis presentation, I want to frame gluconeogenesis in the context of the central metabolic pathway diagram. At the top, I'm showing in blue glucose to pyruvate glycolysis. In the middle in purple, I'm showing the conversion of pyruvate to acetyl coenzyme A, which feeds into the citric acid cycle, the circle in the middle in black, and ultimately electrons fall into the electron transport chain in oxidative phosphorylation. Another way of thinking about how to fit together the different metabolic pathways is in the context of a figure of the cell. Specifically, glycolysis all occurs in the cytosol down to the production of pyruvate. Pyruvate, the oxidized product of glycolysis, gets pumped into the mitochondrial matrix, where it can enter the citric acid cycle by being oxidized to carbon dioxide in order to acetylate coenzyme A. Acetyl coenzyme A enters the citric acid cycle, which produces a lot of NADH from NAD+, which is high, which is low in energy. That all occurs in the matrix. NADH then binds to transmembrane complexes of the electron transport chain and dumps those high energy electrons into those complexes to fall down the electron transport chain and ultimately to drive oxidative phosphorylation to make ATP. And since these complexes are all transmembrane, and since these complexes interact with substrates both in the matrix and in the inner mitochondrial space, oxidative phosphorylation is best described as taking place in the inner mitochondrial membrane. And right now, I want to go back and look at glycolysis and gluconeogenesis. I mentioned that glycolysis oxidizes glucose to pyruvate. How many ATP do you get from that? You get 2. Right now I want to look at the numbers. So we know that the hexose phase is the investment stage. You're putting money in. How much ATP does the hexose stage cost? Well, 1, 2. You burn 2 ATP. You're in the hole. You've borrowed money to start your business. So now let's see if we can recoup our investment and if we can make a profit. And that'll occur in the trio stage. So let's see. We have 2 steps named for the reverse direction. I hope you know these enzymes from the glycolysis talk: phosphoglycerate kinase and pyruvate kinase. They're the only kinases in the trio steps. And I'm saying, wait a minute. There's only 2 ATP in the trio steps? Well, we spent 2 AP in the hexoses steps. We're not going to make any profit. Of course, there's a flaw with this reasoning. How do we get a profit? Please pause and answer. It's pretty important to understand. Well, of course, there's 2 trioses for every 1 hexoses. So in glycolysis, to prepare the hexoses, you do burn 2 ATP. However, before you recover ATP, the hexose is cleaved into trioses, so there are 2 trioses per hexose. Then, oxidation of each of those trioses produces 2 ATP each, making a total of 4 AT produced, 2 ATP invested to give a net of 2 ATP gained. And I'll say, that's okay, but it's not real impressive. What's going to be real impressive is an oxidative phosphorylation where we not only have the 2 that we get from glycolysis, but about 30 more. Why do I say about 30? It literally depends on exactly how the pathway is followed and the imperfections in the pathway. It's good enough to say that complete oxidative breakdown of glucose from glucose to carbon dioxide and water gives around 30 ATP. You want to be very specific that glycolysis itself produces 2 ATP. Most of the steps are reversible, as indicated by these blue double-tipped arrows. Only 3 of the glycolytic enzymes catalyze irreversible steps, shown in red, and they're irreversible because they represent a big drop in energy. These 3 irreversible steps are, of course, also the regulatory steps. So, for example, hexokinase in phosphorylating glucose to make glucose 6-phosphate is irreversible and regulatory. That is, glucose 6-phosphate is a substrate of glycolysis, so as it builds up, it will tend to be driven down the pathway. However, hexokinase is not the committed step to glycolysis because glucose 6-phosphate can also enter glycogen synthesis or the pentose phosphate pathway. In contrast, phosphofructokinase 1 is not only a regulated step in glycolysis, but it is the committed step because fructose 1,6-bisphosphate cannot enter into glycogen synthesis or pentose phosphate. It can only go down into glycolysis. Finally, pyruvate kinase is also a step which involves a drop in energy, and so it is irreversible, which means that pyruvate will not be able to just flow back into glycolysis, but it must go into a different pathway. One of those alternative pathways is the citric acid cycle. Pyruvate can enter the citric acid cycle, and this links glycolysis to the citric acid cycle. Briefly, pyruvate dehydrogenase grabs pyruvate and removes a hydrogen, producing carbon dioxide to power the acetylation of coenzyme A, which can then be bound by citrate synthase in order to donate its acetyl group to oxaloacetate to make citric acid. Today I want to talk about a different pathway, and that is where instead of going into the citric acid cycle, pyruvate goes into gluconeogenesis. Whereas glycolysis oxidizes glucose to pyruvate to make ATP, gluconeogenesis takes pyruvate and rebuilds glucose at the expense of ATP. Accordingly, each of those negative energy steps that was regulatory in glycolysis, shown in red, must be reversed by a different negative energy step, shown in green, in gluconeogenesis. For example, pyruvate kinase in gluconeogenesis must be reversed by an alternative pathway which begins with pyruvate carboxylase. Interpret the name to give me some idea what it does. If pyruvate has 3 carbon atoms in it, how many atoms will there be in oxaloacetate? Pyruvate carboxylase acts on the substrate pyruvate, and it adds or links a carboxylic acid to it, so that does add a carbon atom to the 3-carbon pyruvate to make a 4-carbon atom oxaloacetate. The next enzyme in this pathway, phosphoenolpyruvate carboxykinase, is going to form phosphoenolpyruvate by burning the carboxylic acid off of oxaloacetate to power the phosphorylation, so that we have phosphoenol pyruvate, a 3-carbon intermediate with a phosphate group. As the concentration of phosphoenol pyruvate builds up, and assuming the glycolytic enzymes in red are inhibited, pyruvate kinase, phosphofructokinase, and hexokinase, what then will happen to the concentration of 2-phosphoglycerate, 3-phosphoglycerate, 1,3-bisphosphoglycerate, glyceraldehyde 3-phosphate, and fructose 1,6-bisphosphate? Please pause and answer. This is a critical question. These are all reversible steps. All these intermediates will just flow back and forth in equilibrium such that all of these intermediates will be in high concentration, including fructose 1,6-bisphosphate. And of course, now we're at that critical next regulated step. At this regulated step, if the green gluconeogenic enzymes are inhibited, and if the red glycolytic enzymes are activated, phosphofructokinase 1 will add a phosphate to fructose 6-phosphate to make fructose 1,6-bisphosphate. Since, however, we're looking at gluconeogenesis, and we can assume the red glycolytic enzymes are inhibited and the green gluconeogenic enzymes are activated, fructose 1,6-bisphosphatase will act upon fructose 1,6-bisphosphate, and it will cleave off the phosphate to convert fructose 1,6-bisphosphate to fructose 6-phosphate. Again, fructose 6-phosphate can flow back and forth with glucose 6-phosphate, and for that matter with glucose 1-phosphate. Regardless, as glucose 6-phosphate builds up, it can be acted upon by glucose 6-phosphatase, and that's an enzyme again that we described back in the glycolysis talk. Glucose 6-phosphatase, which will break off the phosphate to regenerate glucose so that a liver cell could export glucose made from gluconeogenesis. Before I look at a little more detail at a couple of these regulatory enzymes in gluconeogenesis, I thought I would look at some numbers. How many ATP do we get if you burn 1 glucose to pyruvate? And then the other question would be, if we take 2 pyruvate molecules to regenerate glucose, how many ATP does it cost? And I have to point out that there's always a cost of doing business. Hopefully you remember that we get a net of 2 ATP per glycolysis. Hexokinase costs 1, phosphofructokinase costs 1, phosphoglycerate kinase and pyruvate kinase each return 2 times 2, so we know there's a net of 2 ATP you get from glycolysis. How about gluconeogenesis? To be clear, any business knows that you never get back what you put in. There's always a cost to business. So if you're going to get ATP from glycolysis, to go back and rebuild glucose will cost more than a net of 2. And in fact, if we take a look at it, here I'm adding the ATP to the equation, hexokinase minus 1, phosphofructokinase minus 1, phosphoglycerate kinase plus 1 times 2, pyruvate kinase plus 1 times 2. So as I mentioned, glycolysis gives us a net of 2 ATP. Now if we look at gluconeogenesis, pyruvate carboxylase costs 1 ATP times 2. Phosphoenol pyruvate carboxykinase costs 1 times 2. Phosphoglycerate kinase costs 1 times 2. That's 1, 2, 3, 4, 5, 6. So whereas we can get 2 ATP from glycolysis, if we're going to be running a cycle, which our bodies do, you're going to be costing 6 ATP every time you make 2. So there's a loss of 4 in order to be using this type of a cycle. Now I would like to look just a little bit at regulation. To be clear, if you're going to activate glycolysis, you will inhibit gluconeogenesis. If you're going to activate gluconeogenesis, you're going to inhibit glycolysis, because if you are running both cycles at the same time, you're going to be burning and burning and burning ATP, making a bunch of heat, but you won't be getting any ATP. This is called a futile cycle. To avoid a futile cycle, you will need to inhibit the red glycolytic enzymes when you activate the green gluconeogenic enzymes. And you'll need to inhibit the green gluconeogenic enzymes when you activate the red glycolytic enzymes. And in fact, I'd like to try a simple example of regulation. Here I'm showing glycolysis to the left, gluconeogenesis to the right, and I'm showing the key regulated enzymes, phosphofructokinase and pyruvate kinase. Each of these enzymes is allosterically regulated. Allosteric means other site. For example, ATP binds to phosphofructokinase in the active site and is a substrate. But in addition, there's an allosteric, different site on phosphofructokinase that binds to ATP, and the binding to the allosteric site induces a conformational change that affects the activity of phosphofructokinase. Now, based upon your understanding of glycolysis and gluconeogenesis, can you tell me whether or not ATP will stimulate phosphofructokinase activity, or will it inhibit phosphofructokinase activity? The same for AMP, the same for ATP's effect on pyruvate kinase, ADP activity on phospholynyl pyruvate carboxykinase, and on pyruvate carboxylase activity, and the effect of AMP allosterically regulating fructose 1,6-bisphosphatase activity. Spend a couple minutes thinking about this, but if you're finding yourself getting bogged down, on the next slide I have some clues. Most cells need to regulate their own internal energy even independently of what's going on outside of the cell. And what is the purpose of glycolysis? It is to make energy in the form of ATP. Therefore, how do you think an excess of ATP will affect the activity of phosphofructokinase? Based on that, again, try to answer as many of these as you can. Nonetheless, I'm going to move on. In any cell, if you have excess ATP, that will be a signal that, hey, we don't want to run phosphofructokinase because we already have a lot of ATP. Why would we make more? In fact, you can have too much of a good thing. In contrast, if you have a whole bunch of AMP, that's a signal that you're really low on energy. So AMP binds to an allosteric site on phosphofructokinase, and what does it do? It will upregulate it. AMP says we need ATP, so it will upregulate the committed key regulatory enzyme for glycolysis. How about pyruvate kinase? Pyruvate kinase is the last step in glycolysis. It is the enzyme that actually forms ATP. So if we have an excessive amount of ATP, we do not want to run pyruvate kinase to make more ATP. High energy will downregulate glycolysis, and low energy will stimulate glycolysis. Now, in gluconeogenesis within a liver cell, I'm only showing AMP and ADP. How do you think they will affect gluconeogenesis? Please pause and answer. Well, you really can't make glucose unless you have energy. If you have high ATP in the liver cell, you will be able to run gluconeogenesis. If AMP activates gluconeogenesis and it also activates glycolysis, you're running a futile cycle. So just as AMP stimulates glycolysis, it will downregulate gluconeogenesis. How about ADP? Well, you're saying that's between ATP and AMP. And it is. AMP is a sign that, wow, we are really low on energy. But ADP is also a signal that energy is low. So accordingly, ADP is a sign of low energy. In low energy, you will have trouble running gluconeogenesis, so that will downregulate pyruvate carboxylase and phospholynyl pyruvate carboxykinase to slow down gluconeogenesis. I think that when I describe enzymes purely based on language, it might be hard to remember them, whereas when I describe them with language and also include showing you structures that tie in with the language, it might make it easier. In fact, I want to take a look at pyruvate carboxylase. That was the first enzyme in gluconeogenesis. And I want to look at the structures of the substrates. And I'd like to begin with pyruvate itself, since pyruvate will be the substrate of pyruvate carboxylase. If you look at this figure where they actually show every carbon and every hydrogen, certainly that's more complicated than the way I show it. Nonetheless, you do have the carboxylic acid group, you have the ketone, you have the methyl group. And here again, the carboxylic acid, the ketone, and the methyl group. Since pyruvate is going to form a carbon-to-carbon bond with a carboxylic acid group, that carboxylic acid group has to be in an activated form. You can't just grab carbon dioxide or carbonic acid. And in particular, that carboxylic acid group is activated by linking it to biotin, one of the B vitamins. Once you've added the fourth carbon to the 3 carbons of pyruvate, you form oxaloacetate, and this is how they show it. Again, I think it's fairly complicated. I think that this figure is a little easier to interpret. And in fact, if you look at the black part of oxaloacetate, the black part corresponds with oxalic acid, which would have a fourth oxygen where there's the carbon over here. And so oxaloacetate is the name of that compound, and this is the way they would show it. Oxaloacetate can be reduced to malate. I know it's reduced because you take NADH and oxidize it to NAD+. Good to know what that means. But malate then is exported from the mitochondria, where it can be converted right back to oxaloacetate. Why are they doing that? Because part of gluconeogenesis occurs in the mitochondrial matrix, and the rest of it occurs in the cytosol. Glycolysis occurs completely in the cytosol, citric acid cycle completely in the matrix, oxidative phosphorylation completely in the inner mitochondrial membrane. Since gluconeogenesis is mixed between the mitochondrial matrix and the cytosol, I just didn't want to make it that complicated, but here we have phosphoenolpyruvate ready to be acted upon in gluconeogenesis by phosphoenolpyruvate carboxykinase to do the next step. Well, that's as far as I want to go with gluconeogenesis. I know there's some parts that are a little complicated there, so I hope if there are parts you didn't understand, you won't hesitate to call me. I'm going to try to be available over the whole weekend and all. The final is next week, so good luck and thank you.